morning, everybody, and welcome to the 22nd Food Safety webinar entitled Lateral Flow Immunoassay for Rapid Detection of Food Pathogens and Other Contaminants. Our resource speaker for today has a PhD in plant pathology, major in virology and immunology, and minor in microbiology from Iowa State University, USA. She was granted with various scholarships from Bayer Chemicals Philippines, IPB UP Los Baños, and USAID. She is currently a faculty at the University of Phoenix, USA, teaching science-based courses at the College of Natural Sciences. But from March to June of this year, she is one of the DOST Balik Scientist Awardee for the project Development of Lateral Flow Immunoassay Technology for Plant Virus and Foodborne Pathogens, which is being hosted by Biotech UPLB. Ladies and gentlemen, Dr. Angelita Talens.
Good morning. I'm honored, really, indeed, to be a part of this food safety interdisciplinary or interagency program. I didn't have any hesitation to accept this, so here I am. I wish I could have a longer one. Like the title is actually including other contaminants. If I will include this, it will take more than 1 hour. So what I'm going to do today is just to show you what are the sources of foodborne poisoning or pathogens, and later on I will talk about how to detect them or identify them. And so there'll be two sets of my slides, which I hope you'll be able to grasp what I am going to share with you today. So let's start. As you can see, my first slide is the tulips. I come from Seattle, Washington. It's Washington State. It's the northern part of the west side of the USA. It's in fact above California, so it's really cold up there. And we have tulips every April, and I'm just showing you this one. So I just love these beautiful flowers, which we cannot see here in the Philippines. My first slide shows you about the foodborne illnesses, and it's just a simple definition. Is any disease or illnesses resulting from eating contaminated food items. So if you go to the next slides, you will see the other how the foodborne pathogen will enter into the food chain. And I have, of course, I'm sorry, I'm coming from the US, so I have the map of the USA in here. So you can just see it's globally already. The food supplies enters globally, so you can see how it's easier to bring in into another countries or even the Philippines some foodborne or food contamination. In this next slide, you will see the multiple points of how a pathogen will enter into your food chain. Number one, of course, production from the farm. You know, all the foods are coming from the farm, right? And then next will be the processing. Next will be how they are being distributed and how they are being stored. And I hope the way you store the food in here will be the same thing like I've seen, because I've been staying in the US for like 30 years actually already. So I hope we are doing the same thing in here. And of course, transportation. We also have some problems in there. It's, you know, sometimes they use trucks that are not refrigerated maybe, so that will be a cause of food contamination too. And also how they're being sold in the market, open market or closed market or supermarkets, how they're being prepared, home cooking or you eat outside, which normally now I see a lot of people eating outside. I've never seen those many people eating outside already. And how it's being consumed. So next slide, please. And then this will show you where are the sources of contamination or what agents are contributing to the food contamination. As we can see, of course, the air, the soil, water, also the ingredients of the food that you're eating, especially those processed foods, and how they're being packaged. And of course, the vermin. When during storage, you see a lot of rats and other things, cockroaches. Also, and also the surfaces where the food is, you know, having a contact with, whether it's a clean surface or not, and people who are handling them too, whether they use their dirty hands or gloves. Next slide show you like how do we know for sure that we are contaminated or we are infected by any kind of food poisoning. As you can see, the slide shows you different people from different areas, but as you can see, the main thing is you see the symptoms or the signs, usually nausea, fever, vomiting, pain in your abdomen, and causes diarrhea. And on the left, these are all the different morphological attributes of the pathogen that will maybe cause vomiting or any of those symptoms that you see in here. Next slides show you. I have a map. Again, I assure apologies that I'm using a U.S. map here, but I just want you to know that in the USA, this is 2012 actually. As you can see, let's see, I will show you. This here up here is all the total outbreak from viruses and bacterial causes, and this is where I'm coming from, the Washington State. Oh, I'm sorry. Well, let me see if I could go back. You can see the darker the oops sorry the darker the areas or the state, that means they have higher incidences of the outbreaks. Okay, never mind. I just wanted to show you where I'm coming from, but it didn't want to stay. The next slides show you the Center for Disease Control in the USA. It's just a 2011 estimates. That's all that I got from the internet, and they say that 1 out of 6 Americans are getting sick, or equivalent to 48 million people. And out of this, 128,000 were hospitalized because of food poisoning, and 3,000 died because of the food that they consumed. So that's how they estimated from the Center for Disease Control in the USA. Next slide show you again, out of the 5 top, 13 specified and unspecified agents that will contribute to the acquired foodborne illnesses, domestically acquired. Most of them are from the group of viruses called noroviruses. Second will come from salmonella. Usually you get this from chicken or from the eggs. And the third will be Clostridium perfringens and Campylobacter, usually from fried chicken. You can get this. The Staphylococcus aureus you can get a lot from, even from the water, from your milk, things like that. Okay, so the next slide will show you again, out of the top of the 31 specified unspecified agents, the following are requiring hospitalization. Again, number 1 is from a virus group, noroviruses. Second is salmonella again, Campylobacter, Toxoplasma. These are coming from, and number 5 is Escherichia coli. Actually, this is the so-called 0157. I think this is also ongoing here in the Philippines. I noticed that there are a lot of E. coli also in some of the food that you can consume from the outside. Next slide will show you the local condition. I was fortunate enough when I was browsing the internet, I saw some reports coming from here, and between 1995 to 2004, there were only 60 outbreaks. And I thought 60 only? Maybe it's because they don't report it, so they only have a very small amount. But look at this. It's very interesting. Spaghetti. Why? People love spaghetti and they reported the outbreaks from mostly from the food spaghetti being served as a food vehicle. And normally they were able to get this 60 incidences from food services, from school, workplaces, and they found out they have identified number 1 cause is salmonella. Next you'll see a Vibrio, staphylococcal enterotoxins, and also shellfish poisoning, and histamine. It's just an allergenic reaction, histamine. Another one was in 2013, coming from Sunstar from Baguio, reported by Department of Health Assistant Regional Director Amelita Pangilinan, and she reported 1100% increase in bloody diarrhea, and around 50% of this increase was in hepatitis A in the Cordillera region. So these are the only listed information that I got locally. Another one is in 2014, reported by Vital groups, and they sampled 300 food samples, and they were able to be identified as positive for E. coli, around 16.7%, and Salmonella, 24.7%, and for somatic phages, 47%. This is very interesting. There is no significant differences whether you get this food from the open market, which is very, very, very common here. We call it the palenque, and also supermarkets. So it doesn't mean that if you go to supermarkets, you are getting clean food. So You also get those in fact contamination. These slides, okay, please go to the next slides. These slides will show you different types of morphological shapes and sizes or forms of the different pathogens that are contaminating mainly our food. We call this Campylobacter, the viruses. These are all viruses. Uh, these are the agents that you can see from using your naked eye. But bacteria, sometimes you can see some signs better than the viruses. Salmonella, of course, and then the 0157. This is one of the famous contaminants in the USA, 0157, which contaminated a lot of those hamburgers being served in chain restaurants. You also have the Clostridium, and these are parasites, Giardia and Toxoplasma gondii. So these are just some samples. There's a lot of different pathogens in there that are being contaminating our food supplies, but these are just some examples. The next slide will show you sources of contamination. They could be biological, and these are bacteria, viruses, prions. Do you remember when I think, I don't know what year was that, when a lot of Filipinos like the smiling spam or luncheon meat? The prions, we call it prions because it was called the BSE or mad cow disease. So if you can remember that, that was good. Those are the prions. And parasites also are biological sources of foodborne illnesses. These are the causative agents, or a combination of this, that will cause these illnesses of people consuming contaminated foods. Next slides, we continue on biological agents. Pathogenic bacteria examples are the E. coli or Escherichia coli, and usually, very commonly, is the 0157H7. There was a lot of scare in here in the USA before because of this in hamburgers. And they could be, you don't have to know all the second EHAC. Those are just different strains of the E. coli 0157. Next one is Listeria species. Next one is Salmonella. Bacillus also. Shigella. This causes toxin. Vibrio. Vibrio normally comes from shellfish, oysters, mussels, or whatever you can get from the sea. Those if they are infected with Vibrio, then that's contaminated. Campylobacter, again, very commonly in fried chicken. Clostridium. Next slides, more biological agents. We have the viruses now, enteroviruses, hepatitis A and E. E is not really common in the USA. I think it's available here, or it's been identified in the Philippines. Noroviruses, we have the O and the non-O. These are just strains. Rotaviruses, prions, like what I've said, the maling, luncheon meat, BSE, bovine, spongiform encephalitis. So it's just like a prion. It's not a virus, but it's also a cause of it's also biological agents that will contaminate even a food that is already processed, like maling. So, okay, next slide, please. We have other biologicals that are listed in here, like the so-called parasites. Number one is Giardia. As you remember, I've shown you previous slides. These are the flagellated protozoans causing giardiasis. They call those disease. Others are Sarcocystis cruzi, another protozoan. Toxoplasma, it's causing toxoplasmosis. That's the name of the disease. Fasciola hepatica is a parasitic flatworm that could contaminate your food, and then when you eat it, it goes into your system, into your intestine. Another one is the anaerobic protozoan Entamoeba histolytica. And also the trichinella is a nematode parasite. So these are like worms or one-celled pathogen. I have a slide. The next one will be the guidelines from the Center for Disease Control in the USA. How do they detect foodborne pathogens like the E. coli? They usually do isolation, just the plating, simple plating. So they isolate the E. coli, and so mostly all the bacteria, they use plates to isolate. And on my left, as I point the cursor in here, you can see an E. coli, very beautiful cells, or it grows into the colonies into the in the plate. If you're interested about, I mentioned about this EHEC, E-Tech, E-Pec, E-Tech. These are all different kinds of E. coli. Now, the next slides will show you other pathogens like Bacillus cereus, and the way they detect it is if they find around 10 to the 5 colonies of Bacillus per grams of food samples, then they will call that, it's causing the disease or causing the foodborne illness. The Campylobacter, if they can isolate implicated food from 2 or more peoples, then that's a cause of alarm already. Then they will usually go back to where the food source coming from, and they usually shut down. Like in the case of E. coli before, Jack in the Box, I think 1983, there were quite a few people who got sick from eating on hamburger. So FDA went to the Jack in the Box and they just shut it down because they were able to see and isolated E. coli and other kinds of pathogens there. Clostridium again, 10 to the 5 per gram. So if they find this, they find number of colonies like this in the food sample, then that's a cause, that's an alarm. Of course, another one is Listeria monocytogenes and Salmonella. So normally they just use a simple plating using blood agar or brilliant green agar, and they will count the number of colonies from there. Vibrio and Yersinia, that's the next slide. Next one will be the viruses. Viruses are hard to isolate, but usually what they do is they take your blood and they will just detect or find out if you have elevated antibody response. This is the immunoresponse of a human being. Once you get infected with viruses, your immune system will produce antibodies against these viruses, and they will test you. If you have an elevated antibody response, then you're pretty sure that you are being infected with a kind of virus contamination. Same thing with the HEPA A and B, and also so all kinds of viruses, they need to be isolated. They need your blood. They take your blood and do some testing about antibody response. And I will mention that later in my next slides. Other forms of identifying how you are infected. This slide, the next one will just be a simple diagram. It's a very simple one, and that we do as microbiologists usually. You just do the gram staining. I hope you're familiar with gram staining. So if you see that it's positive, then it could be if there are rats or cocci, then you do all the following tests: hemolysis, oxidase catalase, some biochemical test. And if they're negative on the gram staining test that you did, and the both are rats and cocci that you see under the light microscope, then you also do this. If they are positive, all these different tests, then you do more identification. They're usually a book, so you know what species or what kind of organisms that are infecting your food. Next slide. Another way to identify that you have a foodborne pathogen is based from the Food and Drug Administration in the U.S. So they have rapid methods that they are using to detect the presence of foodborne pathogens. The first one is just a biochemical kit. So they have a lot of kits already. I'll just show you a list, but you don't have to remember all of this. These are just, list of different detection mechanism or tests that they're using in the U.S. And a lot of companies are already have this. They are producing this, manufacturing this. They even do the MIS already, or the micros can walk away. They're, all the names are really very interesting, but they're all being used. Next slide, another set of methods. You have PCR. I think you're familiar with this. This is a polymerase chain reaction. It's the genomic-based kits, and they use the nucleic acid, DNA, or the RNA composition of the pathogen. And Biocontrol is a company who developed this Probella PCR. They use it specifically for Clostridium botulinum. That's the one that causes poisoning, when you eat a canned food that is infected or contaminated. Neogen is another very popular company in the US. They produce PCR BACS kit for E. coli 0157. So you name all the different food pathogen contaminants, and you can get any kits in the USA already. But these are all the genomic based or nucleic acid based. Salmonella. Next slide will show you other methods that are genomic based again. They use probes, is the DNA or the RNA component of the pathogen. And again, you see Neogen. Neogen is really very popular. You can find it anywhere. And they have, again, another kind of probe using an AccuProbe for Campylobacter specifically, and also for Escherichia coli. It's 0157 and Listeria. So Neogen is all over the place. Salmonella. So if you have problems for, like you think your food is contaminated with any of these pathogen listed, just go online Neogen and you can get all these kits. They have all these kits available. Neogen. Next slide will show you other kinds of methods again to detect. And this is where I'm coming. Antibody-based detection kits. Remember when I talk about the antibody when you are infected with noroviruses or any kind of viruses that contaminate your food, you go to the clinic and have your blood tested. So with your blood, they will see a raised antibody reaction or response. That means you are infected. So they're using antibody. And Tecra is another company that are already using a kit against Bacillus cereus. They are using ELISA. ELISA, I hope you are familiar with this. It's been since 1977. They've been using this. It means an enzyme-linked immunosorbent assay. They use enzyme to conjugate. They bind it with the antibody, and they use that to pick up the virus or whatever kind of pathogen that they could pick it up. So Tecra, Unipath also use, this is not ELISA, but it's also antibody based. It's like agglutination. So all they need to do is just add the sample, maybe your blood, and they add the kit or the antibody based kit, and there will be agglutination, or you can see precipitation in the mixture. BD again uses the latex agglutination. It's similar with Unipath. And others, there are a lot of them using ELISA kit and latex agglutination for different kinds of toxins. Okay, another one that they're very popular right now is the use for another antibody base, but they use strips. As you can see this picture in here, let me see if I could, okay. These are strips. This is where I'm coming from. We have the lateral flow, but this one, they're also using a strip, but they call it dipstick. The kit should come along with different kinds of reagents. So you will just add your sample into the kit, and voila, you will have positive, negative. And this is one that I came here for because it's like really very fast, rapid, and it's a point of use. So it's a device that it's a chromatographic device, and I will show you in my next slide. Okay, here it comes. I call it lateral flow it's a chromatogram kind of thing, assay, or I call it LFIA. Excuse me. This test actually was inspired by a pregnancy test, and I think I know you know what I'm saying. All women or even men, they go buy this pregnancy kit or test kit from a pharmacy. It's out of, you don't have to have a recommendation or a prescription from your doctor. So this is the same thing. In 1960s, this was the first thing that they did. They were able to, Margaret Crane was able to develop a kit that uses agglutination kit by using the human chorionic gonadotropin hormones from the woman. So if you are pregnant, then you will be positive with this. And 10 years later, it was approved by the FDA for use in the Europe and US market. Between the 1980 and 1990, they expanded this to be available to the home market. So now any woman who would like to be pregnant or scared of being pregnant can go to the pharmacy and just pick this up, and you can just add your urine and find out if you're pregnant or not. But in 2003, they extended it into a digital pregnancy test. Now, the kit will show the word pregnant or not pregnant, so it's easier. They don't have to know how to interpret the kit, but now they can see the word pregnant or not pregnant. So this is the one that we took into consideration and develop the lateral flow test. And characteristics of the lateral flow test should have the following. Should be accurate, should be easy to use and interpret, that anybody can pick up, can use it without going to the school to learn about it. Should be at least not expensive. Should be easy to use. You can bring it anywhere. Stable. It can last maybe 1 to 2 years. Of course, it's acceptable, and you can produce in mass quality or quantity. Yeah. Okay. The next slide will show you. These are the ideal point of care test should have the following characteristics. It's called assured. So you have to assure your client that the test that they are buying from you is really good. So should be affordable, sensitive, specific, user friendly, hindi nakakatakot. It's easy to use, rapid and robust, and you don't have to use an equipment to be able to use it, and it could be delivered to any end user. Next slide. I'll show you how this is being done, or what is this all about. So I'll show you a little bit about what is the concept or the principle, and the next will be how to use it and who are using it, where can you apply it. So the LFIA concept is this. The next slide you will see a set of 3 different things. You have on your left, you have the antigen. So your antigen can be your bacteria, which is the 0157, or viruses, Norwalk virus, and you have this Y molecule. We call this the antibody. So the reaction will be, if this will be, it's just to react, and you will see the Y connecting to one of the we call it epitope or antigenic determinants of your antigen. You only see one in here because it's monovalent, monoclonal. It's only reacting to one. As you will see later on in my other slides, that there are different forms of antibody. I would like to point this out on the lower part. If you can see the high specificity, you have the lock and the key. So the interaction of the antigen and the antibody is just like a key and a lock. So if your lock is not specific to your doorknob or the key or whatever, it does not open, then that means it's negative. The same thing with this. So just remember this. This is how I usually teach my students so that they will remember what is the antibody antigen reaction. It's like a key and a lock. If they don't connect, or if they don't open the lock, then it's not specific. Can you remember that? Because I'll be mentioning this also in my next slides. Okay, next slides. Okay, we talk about the concept antigen. The first reagent for your kit is the antigen. The antigen could be biotic, plant, human, or animal, bacteria, viruses, fungi, genetically modified organism identifiers, like you have heard maybe the BT eggplant, BT corn, right? Or biomarkers for some cancer cells or others. So those are biotic. Antigens can also be abiotic. It could be allergens, could be toxins like aflatoxins. Maybe you have heard about aflatoxins from the corn or from the coconut, copra. So those are toxins. Could be also antibiotics. Oh, okay, I'm sorry. Let's go back. So could be drugs, maybe shabu, or what is the popular in the Philippines? Marijuana. But fortunately, in where I'm coming from, it's legal to take marijuana. So you can come and eat something else from marijuana. Pesticide, herbicide. So you name it. These are the abiotic antigens. But you can also produce antibody against all these different abiotic. Next slides will show you the antibody. So you remember the concept, the 2 reagents. Remember the antibody, the antigen, and the antibody. Antigen are the viruses, bacteria, aflatoxin, and you name it, okay? And the antibody, which is specifically produced against the antigen, could be polyclonal or monoclonal. The first previous slide that I've shown you is the monoclonal. Okay. Mono means one, right? So it can only react one antigenic determinant of your bacteria or your viruses. Poly means many. Where's my cursor? There. Poly means many. So if you remember my slides before, there were different kinds, like there's a round, there's a triangle, heptagon, on those antigen, and that means those are the different kinds of antigenic determinants. So if you produce a polyclonal antibody, It can react with all those different antigenic determinants. And you can produce it from rabbits, from goats, sheep, horses, and even chicken. Next slide will show you the reaction, how they look like. So if you are developing the diagnostic kit using the lateral flow or any kind of immunoassay or antibody based, your goal is this zone of equivalence, so that you will see a precipitation form. If you have a more or excess antibody, which are the Y, you will not see anything. If you have excess antigen, which are the round ones, you will not see anything. So your goal, if you're developing a kit, should be here, zone of equivalence. Next slide will show you practice. Now we go to the practice. We're done with the concept, antigen, antibody. Now we go to the practice. How is it? How can you make a strip or a lateral flow kit? This slide will show you all the different parts. You have the plastic backing, you have the sample pad, then you have the conjugate pad, you have the migration membrane where you put your antibodies in here, and you have the absorbent pad or just the wick to absorb the excess liquid. Next slide will be showing you the different parts in detail. So the first part that is very important in this lateral flow immunoassay kit is the sample pad. Sample pad, this is where you add your sample, which is your antigen. So it could be composed of cellulose, a fiber, rayon, polyester, or any other kind. So the next slide will show you the next component, which is the conjugate pad. See, as you can see the color, this is the one that is the most important part in here, because you're putting something else to be able to visibly detect the presence of your pathogen. And this is composed of, could be colored dyes, but I use a nanogold. With the next slide will show you the one that I use. I used a nanogold, or we call it tetrachloroauric acid, if you want to, the chemical name. And they come in different sizes, as you can see, smaller to bigger. So this is only 5 nanometer in size. That's why they call nano, because they're really very small. And 30, next one, then 60. Where is my cursor? Okay, I use usually in between this one, 40 nanometer for my case. And that's the one that I use for my E. coli. So it's in between this color. So as you can see, the tube, the bottles or the vials, they're containing the gold, but different colors, right? So if you see this in between, I use this 40. So if I see like deep red or something like that, then that's the size that I want to use. And I prepare my own gold, but you can also buy it. And the size is down below here. So it's in between this one. So it's not so big, not so small. Next slide will show you the third part of this strip is the nitrocellulose membrane. Usually you can use just a nitrocellulose membrane, a simple one, or a polyvinylidene fluoride. So this is where you immobilize your antibody. These are your antibodies, okay? Capture line is your specific antibody that you produce against your bacteria or against your antigen. And this control line is another antibody that's supposed to be there, but it's not specific. It will always be, it should always be light, like, produce this color, so it will validate your test. Okay, the next slides will show you. The next part is absorbing pad. Excuse me. Absorbing pad. This is where your excess fluid coming from your sample that goes like that. That's why it's called lateral flow, because it moves laterally. Okay, so it will be absorbed in here. So this is just a wick. It's a high density cellulose material. Next slide will show you the whole thing again. So the different parts. This is the strip that I've shown you earlier, which you can also use as is, or you can put it in a housing, plastic housing. Like your pregnancy test kit, when you buy it, is in a plastic housing. So all the different parts in here. Next slide. Okay, now we go to, how does the lateral flow immunoassay work? Remember the strip. So it composed with the different things like sample pad, conjugate pad, and the membrane, and the absorbent pad. So you add your buffer or your sample. This is where you add your sample, right? and then your whatever analyte that you are testing, it will move to the next part of the strip, which is the conjugate pad, and this is where okay, here, okay, and then it will be reacting like this. So the liquid, as you can see, the bluish color is the liquid or the sample. The next slide will show you the third step will be your sample, your analyte will be moving on the membrane, which will be attaching already. So as you can see, attachment with the control, it capture antibody, and this is a control on test line until it goes to the wick. So the excess liquid will go to the wick. So it's simple. It's very simple. That's just this slide. The next slide will show you how it's being done. Actually, if you can just look on your right, that's the lowest part, or the left actually. So this is where you put your sample. Where are you? Okay, here you put your sample. So your sample will move like this. So the first one that it hits will be your test line, or your where your antibody specific to your antigen from your sample will react. So it will light up, right? It will produce the color. The color coming from the gold. Remember the gold? You make the gold, right? And then the next, the whatever is not attached in here will go in here, which is the control line. So if you see two lines in here, it calls positive, because this is the one that will confirm or validate your test. Should always be here. So if there's no line on the test line, that means it's negative, right? You only have the control line. So that's the basic what you see. You only see two. If you can remember how the pregnancy test look like, it's just the same thing, two lines. Next slide will show you different kinds of foods and different kinds of organisms that we get infected with. Normally, hamburger or ham or ready-to-eat meats, they're always, usually, if they're not properly prepared, E. coli or O157. Vibrio cholera from seafoods like oyster. I don't know here in the Philippines if we eat raw oyster, but in America, they eat raw oyster. And if it's coming from unknown sources, they don't eat it. So they have farms actually there. They're being audited by the Food and Drug Administration to test if they're really clean in processing. Okay, bacillus, usually coming from different ready-to-eat meats, which are very popular in the U.S. Clostridium is staph from ham, Salmonella from eggs, chicken, Listeria from I don't know if I've never maybe I haven't been around, but do you have those sushi bar? Okay, be careful, because if they're not really good, prepared very clean, then you get a Listeria monocytogenes. Legionella from water, Campylobacter from fried chicken. So be sure that you know where you're getting your food, okay? Or better yet, prepare your food at home, so you will not be having any problems as long as you know how they're being prepared. So if you have helpers, then maybe you teach them how to clean their hands and how to process the food. There's a lot of different ways to avoid those unnecessary contamination. The next slide will just show you a flow chart how we produce the strips. So we have the membrane system, the conjugate pad where you put the gold. Remember, the gold will allow you to see positive or negative test, and of course sample. I don't normally pre-treat them. I just usually just dispense whatever I need on them and dry them and assemble and cut them into strips and package them. That's the normal process. Okay, maybe you're asking in your mind, how much will you be able to spend in order to manufacture one strip? This is like a data 2 years ago or 3 years ago when I was still working in that company where I made a lot of this. I made 10 million of strips already for E. coli 0157. So the owner of the company, per strip he will at least pocket $1. So if I made 1 million, I made him $1 million. So he was very happy when he asked me, Angelito, when can you make me $1 million? And I was very new, so I said I have to calculate. So after 6 months, I made him 1 million, and he gave me a big bonus, and I was so happy. So that's beside it. I just make you, just like besides the point, but it's only 5 cents in dollar. I don't know in here, but we will see, because all the materials are coming from UK, from USA. So if I will import all those materials from there. I don't know how much will it cost me here, but there's a way to lower the cost. That's why my next project is to produce locally available reagents. Next slide will show you, okay, you can either, remember those strips that I've shown you earlier, you can either see it with your naked eye, or you can use a high-tech. Well, it's not really a high-tech. Some companies in the States, they usually, the kit will come along with a reader. So you can see here, AccuReader, AccuScan. So you can have an idea how much, so you can quantitate it. It's not only by looking quality, but you can also quantitate how much contamination you have in your food. Or you can also have the goggles or the lenses if you want to be high tech, but you don't need to. You can see it actually with your naked eye. Next slides, I just listed the advantages of having this. If you have these strips in the Philippines, which I don't think you have, because I heard that aflatoxin kit that are being used in universities that I met some people, it costs them 500 pesos per one test, and it's coming from the USA. That's a lot of money, right? So if you can make it like 200 or even 100 pesos, that would be good. Should be inexpensive. It should be simple, rapid, and very easy to use. Another thing is should be stable. Remember what I told you earlier part? The characteristics of a lateral flow kit should be at least stable. And the strips that I have produced, they were not encased or they were not housed in a plastic housing. They were naked. That means they were just strips. And it could last for 2 years, just in a box. At ambient temperature, but of course your ambient here will be 35 maybe or 32 degrees centigrade. There is like 24 to 25, so it can last. Maybe it's higher than it could last for shorter. Should not use inexpensive. The equipment that you use to produce it or to manufacture should be inexpensive, and you don't need any expertise to be able to use this kit, and it's very usable at the point of care. So you can bring it because it's portable. Remember the characteristics that I pointed out before? They are portable, inexpensive, no equipment needed. But if you want, you can have the goggle. I mean the what do you call this? Yeah, the lens or the Accu scan. Another one is this is already a mature technology. Remember the pregnancy test came like 1970s, so it's not really hard to make. And it could also be integrated with a portable reader. Remember that I've shown you, and a portable onboard electronics. Okay, of course, if I tell you about advantages, there should be disadvantages, right? The only thing that I can think about is the sensitivity is not really as sensitive as a PCR, the polymerase chain reaction. If anybody else of you have used this, only 1 mole of DNA, you can amplify it. You can see it already. It's there, right? For PCR. But of course, for this lateral flow, you have to have at least, in my case, for the E00157, I have to have at least 1,000 colonies, 1,000 bacteria, to be able to see it. But of course, you can still test it and make it more sensitive. It depends on how you do it. Another disadvantage is you have to have a qualified reagents. You have to have a qualified pure antigen like the bacteria or the viruses, and you have a specific antibody that was produced against the antigen should be also qualified. If not, then there will be a false positives. That's the only thing that I could think of. There are also new technologies right now, which I just would like to mention it to you. These are high tech, cheap technology. The Alpha Lisa is like ELISA, but it's using the Perkin-Elmer. So these are more high tech, and they're very expensive right now. And again, challenges. What are the challenges in how to control the pathogen? It's hard because globally your food supply coming from different countries. Like, where do we get apples here? We don't produce apples, so you get it from China. Oh yeah, so globally you cannot really control. Sometimes they get in. Like we have a lot of emerging pests and diseases in here, and how they also handle it. Clean handling procedure, and again, there's a lot of food chain already out in the Philippines, so it's really hard, and they're even cheaper than you cook it, right? My husband just told me if I eat at home, it will cost you like this, blah 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 blah. If I eat outside, 70 pesos is good enough for me. Things like that. So, but then be careful again where you're eating, because there are, you know, new strains that will emerge, so it's hard to, and also new ways of transmission. If you can recall the Ebola virus, have you heard about the Ebola virus or SARS virus, even the MRSA? There are a lot of those that are being transmitted from bats or animals to humans, like the Reston virus coming from monkey and it got to humans. So just be careful of those new transmission. So again, my question is, what do you choose? Which one of those do you choose, right? To identify and control them. So the selection and support for the appropriate foodborne diagnostic strategy and program that you would like to develop here will have to focus on one or two that most Filipinos will use at specialized sites or point of use, and it should be affordable. And with that, I thank you.
Thank you very much, Dr. Talents, for that very informative lecture. So, our viewers, do you have any questions for Dr. Talents?
Oh, sige. Okay. Was I on time?
Viewers, if you have any questions, you may send it through our live chat.
Oh, I'd like to announce too, I'm going to give hands-on training and demo in Biotech UPLB on June 16. Limited lang ang slot. So if you want, just call me or call Biotech and I could squeeze you in.
So ma'am, how much will the kit cost per test again?
Oh, 5 cents in the US, based on my calculation, 5 cents. But then I'm not very sure yet. Of course, if you I'm not very sure here yet the costing in the Philippines because I have to find out where will I be getting. I just found out that Diamed, if I gave him a list of the things that I need, he will put 2.5 times more. So if my equipment cost me only $15,000 in the US, it will cost 2.5 more, so that's $45,000. So that's the thing. So I have to find out how much will those be before I could cost them. But I hope, if I will produce local analytical reagents, then the cost will go down. Like my next project that I proposed, it already in the DOC Picard, is to produce one of those reagents, those antibody, remember the test line and the control line? I'm producing one of those locally because if you get it from the US, it's very expensive and also the time constraint. So if I'll be able to produce them locally, oh, better. It will lower down the cost. But right now, I don't have any idea. Abangan sa susunod when I come back. What? Oh, okay.
Ma'am, yung test kit, ano pong klaseng mga food or ah s- ano klaseng food yung siya best gamitin or pinaka appropriate gamitin yung test kit?
Sa US, we use it for ready-to-eat meats,
Yes.
like yung mga luncheon, ham. We also use it for how's that? For peanut butter, all those processed food. We also use
For
the
peanut
peanut
butter,
butter.
ma'am?
Yeah, because the aflatoxin. We also have in the company where I worked before. They also have the aflatoxin kit already. So for the E. coli, listeria, usually from ready-to-eat meats or processed food or cooked foods like the hamburger. You know,
How about, ma'am, if for restaurants, for...
restaurants, yes, they do that in the U.S. Yes, even those produce lettuce or before they ship the lettuces from the farm, they let us know, and then we go there to the sources like Dole or Swift. We will test, like we get some samples and we test them. If they're already free of contamination, then you could go ahead and distribute it to those supermarkets kind of thing. So I guess anything. This kit, just like what I did not mention it, but it has a lot of application. It could be for drugs, for testing drugs. Like I remember, I don't want to say it, but in America, kasi the kids they like drugs. They can just get it anywhere. So one time I saw my son, with friends, and I would, oh, I think they're doing something. So what I did, I went to the drugstore and got a kit for marijuana. It's so easy. Just get a strip and you just swab whatever they're using. It becomes positive. So they're using marijuana. So it's easy. Drug substance abuse. It could be for environmental, monitoring. Could be not only pathogen, but also just like what I said, the non-pathogenic agents. You could also do that.
Okay.
Even ngayon yung GMOs, sa states they are using that. Genetically modified, mga BT, mga genes. I think they don't have anything here yet.
So the steep can also be used in detecting GMO.
Yes. Yeah. As long as it's antibody based, as long as you can make the antibody against that antigen.
Okay. We have a question from Miss Loda Nacional. Are there already available test kits for Vibrio parahemolyticus and Vibrio vulnificus?
In the US, but not here.
Oh.
They're still using I think I went to Diliman, the national, what do you call it? Genome, yeah, that's a genome of sa Diliman. They're importing, I think they're using imported kits or they're using PCR, mga primers. Because if you have, primers are the databases for any use anyway, anybody else, so they could just get the DNA primers in there, and then they use it for PCR so they can detect it, but not a kit yet.
Another question from Miss Wilma Del Rosario. How do food chains monitor the presence of pathogens in their products? Or are there really monitoring being done?
In the US, I know because I've been involved with the environment, because the company where I worked before, it's called Institute for Environmental Health. So what we do, they will because this company was for mga consultants then, and they hire all PhDs. So what will they do if they have some problems like that? They will hire us, and then we will go to the source, like the field, the farm, or where they process it. We make samples. We sample their whatever they're washing, every components that or equipment that the product or the produce will go through. Tinetest namin, we make swabs. So environmental monitoring, we will find out what do they have. So if we found some E. coli or any bacteria in those shoots that they're using, right? And then okay, don't deliver those produce because they're contaminated. They've been through a contaminating machine or something like. They think they do, but I'm not sure here. I've been away for like so many years. I don't know if they have it in here. So hopefully they will have. But
Um, they also would like to know if the test kit could also be used for VCO, virgin coconut oil, for test monitoring of presence of aflatoxins.
oh yeah, yeah, just like what I've said. They have already kit in the U.S. Benguet State University has a product on corn for aflatoxin detection. They buy it from the U.S. 500 pesos per one kit. I mean one sample, just testing one sample. So they do that one. So the aflatoxin for that corn can be also used for the copra or coconut oil. That's why I heard. I was just talking to the mother of our chancellor, Dr. Priscilla Sanchez. She's a food scientist, right? She was just saying that the traders, coming from traders for the coconut products to the U.S. They distribute to the Walmart. Walmart is a big company, right? And Good Foods. No, tinigil daw nila is because nobody can testify that the the injection or the pesticide that they're using for the kukulisa can be, kasi it's systemic eh. Can you testify, can you attest na wala doon sa mga product na pinagbibigyan namin sa Walmart? Nobody can attest that. So that means there should be something else that we should do. We should do something else to check kung yung mga products na in-export sa US are free and we don't have. Kaya naka-close daw. Walmart stopped them from sending cocoa products. Sa US kasi they're starting to like the cocoa water already. They like cocoa water.
A question from Miss Catherine Yupanco. Can we look into the possibility of producing test kits using local resource materials?
That's what I'm looking for, but only the reagents. I can promise the reagents. Remember those conjugates, antibody, antigen, but not the materials. Not unless some. But I know in India has already started kasi nag-source out na kami sa India. So if somebody else is good for developing those, why not? Yeah, we have those nitrocellulose membrane. We have those polyester fiber. If they could produce this, why not? Baka meron nga siguro, they just don't know what to produce yet. Because they make a lot of those synthetic materials here anyway, right? So maybe. But for of course, with me starting to develop, I'll be using what I used before because I don't know how to source this out in here. But in the future, maybe, that will bring down the cost if there will be some people who will undertake that endeavor to produce the local materials.
Just a clarification, Dr. Talents. So there are specific test kits per product. Example, different test kits for salmonella and other tests
Yes,
for other
because
pathogens.
because if you use a kit for salmonella, it will not pick up O157 or E. coli because that bacteria was injected, immunized into the any kind of animal like the goat or the rabbits to produce the antibody. And the antibody that is produced is specific to the bacteria that you injected to it. So, kung di mo siya in-injectan ng salmonella, walang salmonella. So it's specific. Everything is specific.
Any more questions from the viewers? You only have 1 more minute left.
For them, is that what you do? Okay. Well, I suppose so. I don't want to, but if that's the way it is, yes, it's okay. I think it's okay. As long as they don't use it, they will claim that it's theirs. Okay, that's fine. PDF, they cannot change it, right? Ask them if there's anybody else who would like to collaborate, maybe for anything. Hopefully I'll come back, and I would really like to start this for any kind of material.
Okay, ma'am. So they're just thanking you for the very informative lecture. So if
Thank
there
you
are
too.
no more questions, again, thank you, Dr. Talents, for the lecture on lateral flow immunoassay technology, and thank you, viewers, for joining us in this webinar. We hope to see you again in our future activities and webinars. So good day.
Thank you.